Let's talk about DNA, transcription, and translation. The human body needs different types of proteins depending on the action it must perform. The information needed to produce those specific proteins can be found in the DNA. More specifically, in the genes. Genes are made out of nucleotides, which are chemical compounds. The nucleotides usually have regions for RNA molecules. These regions have a promoter region and a terminator region. In some genes, the RNA regions are used to synthesize proteins. The process is called gene expression, through the methods of transcription and translation. The transcription process takes place in the nucleus of the cell. In this case, DNA is used as a base to create messengers of RNA strings. In this process, the RNA polymerase enzymes bind to the promoter region. Then the RNA polymerase unwinds the DNA like a broken zipper. The RNA polymerase then slides across the DNA, unwinding it till it reaches the terminator region, where it separates itself from the DNA. While the RNA polymerase slides through the DNA, creates the messenger RNA string by making complementary bases pair up. When the RNA polymerase reaches the terminated region, it gets released from the DNA, also releasing the finished messenger string. Afterwards, the string gets perfectionated by the spliceomes, proteins, and RNA. The spliceomes get rid of the useless parts of the string. Finally, the perfectionated string leaves the nucleus and gets to the cytoplasm. The string then begins the process of translation. The process of translation occurs in the cytoplasm of the cell. This process consists in taking the information of the RNA messenger string to build a polypeptide. The information on the string, the nitrogenous base, is grouped into code groups, codons, which code for different amino acid types. Just as the genes, there are codons that define the beginning of the process and also codons that define the ending of it. Translation begins with the binding of a small ribosomal subunit to the string, more specifically, where the start codon is. Once the small ribosomal subunit is attached to the string, the transfer RNA molecules bring amino acids. The type of amino acid brought by the molecule is defined by this anticodon sequence. After one transfer molecule is attached to the codon on the string, the large ribosomal subunit binds to the string, creating some sort of rooms for the process to begin. The binding of the two ribosomal subunits is called translation complex. The three rooms mentioned before are the sections E, P, and A. When the translation complex starts sliding, transfer molecules are compatible with the current codon in section A, enter through the opening of the ceiling of the translation complex, and bind to the string. Once the two molecules are inside the translation complex, the amino acid from the molecule in the P section binds to the amino acid from the molecule on the A side. Once they are attached to each other, the complex slides, and now the molecule that was in section P goes to section E, and the one in section A goes to P. As the amino acid from the molecule in section P binded to the one from section A, the molecule from section P is now uncharged, so that when it reaches section E, it will exit the translation complex through an opening on the ceiling, and another molecule compatible with the current codon in section A enters the complex. The process is repeated until the translation complex reaches the stopping codons, where the complex unbinds itself from the string, releasing the finished polypeptide amino acid chain. The finished polypeptide then goes to another organelle where it's required. Thank you for watching. We hope you enjoy our video.